Thank you very much for having me. And I think Debbie's talk was a wonderful introduction for what I have to talk about, which is kind of looking at the human side of autism in relation to environment. I started in this field about 15 years ago, and at that time there was virtually nothing known about autism and its causes, and so I'll lay out a little bit with a real focus here on the air pollution. So Debbie also gave you a terrific introduction to what's in air pollution and different kinds of particles and so forth, so I will not dwell on that. And I will just say, and again, she went through this, that obviously the primary focus around air pollution over the years has been on respiratory effects, but also cardiovascular impacts are also quite well established, and now we're in the era where a great deal of work is happening in neurodegenerative diseases as well as neurodevelopmental conditions. So now just a few words about what is autism, and again, Debbie had a whole slide on this, but I'll just say that the defining domains are the four that are here in this slide: social deficits in social reciprocal relationships in communication, the pattern of repetitive interests and restricted interests, repetitive behaviors, and an insistence on sameness, a kind of ritualistic type of behavior and interests. And then also with the latest DSM, DSM-5 now included as part of the core defining features are sensory sensitivities. So many children with autism or adults don't really like being touched or hugged or that sort of thing. The symptoms are generally present by 36 months of age, and it's a little controversial. It has always been considered, or for a long time, to be a lifelong condition, but there do seem to be people who, quote, outgrow it, or as adults do not appear to have the features at that point in their lives. So it is diagnosed using this Autism Diagnostic Observation Scale, the ADOS, and I'm going to show you a little clip of one of these play observations in a moment. That's really the gold standard instrument, and also the ADIR, which is not an observation of the child, so it's a little bit less than gold in its standardness, but it is a standardized and pretty reliable instrument that establishes the history of the child. So the ADOS is really the snapshot in time right now. What does the child look like? The ADIR is really that early development which the primary caregiver provides. And both of these provide separate scores for each of those domains I mentioned. So we're going to see if this works. And this is an ADOS. The child is about 2.5. The activity here is that there's a little piece of Play-Doh, which is the cake for a birthday party, and the little doll on the left, it's that baby's birthday. And you'll see how the child responds to the psychologist here.
Now we have to light the candles. Ready? Let's light them. Oh, it's hot. Oh, 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 hot. Ready? Let's sing. Happy birthday to you. Happy birthday to you. Happy birthday to you, baby.
At this age, I've been to a few birthday parties. They know the song. He's completely unengaged.
Okay, time for cake. Baby's so hungry.
Playing with the Play-Doh.
Baby's so hungry. Time for cake.
So,
can you give baby some cake? He's hungry.
At this point, he drops the
here you go, baby. Here you go.
That she's cut off of the Play-Doh, and he bends down to picks it up. It takes him a little while to do that, so you don't have to see the whole thing. But basically, he pulls it back up again, and he puts it on the spoon. Oh, actually, no, here it is. And he tries to...
Oh, no, not for you, honey. Not for you.
So he has clearly missed the entire point here that this is a pretend cake. It's not a real cake. So the sex ratio in autism is 4 to 1, so 4 males for every 1 female. which makes all of these sex differences, particularly with the, I guess that was with the postnatal exposures that Debbie just showed us, quite relevant. The prevalence is currently 1 in 59. When I started in this field, I think it was about 1 in 250. And the history of thought about what are the causes has been dominated by some publications that came out in the 1960s by Bruno Bettelheim that basically blamed poor parenting, or the term refrigerator moms became popular. So, of course, we now understand that there is a neurobiological basis, and even in humans, we have postmortem autopsy studies, there's electrophysiology studies, and MRI and functional MRI showing what some of those lesions are, and I think we just saw some really compelling evidence about what may be going on at an even more micro and molecular level. A really important part about thinking about etiology of autism is that it is multifactorial, as most complex conditions are. Most cases don't have a single cause, although for the first 10 years that I was working in this field, I can't tell you how many times I heard talks about, we've found the cause. And it just, it made me sort of laugh and cry at the same time that was the kind of dominant way in which scientists were thinking about autism. So what I have on this slide are these pies, what are called sufficient cause pies, where there's a set of factors that together are sufficient to bring about the condition that you're looking at, in this case autism, and there may be multiple such sufficient causes across the population, and of course within an individual this means that there are multiple causes. So that's sort of just a starting point, and I think that Ted Chetler has sort of laid the foundation here for really thinking about the complexity of the kinds of factors that may be at play for any particular child. So vulnerability and resilience, just a few words about that. There's a couple of papers that I like to cite here. The one by Chirag Patel was looking at the NHANES, National Health and Nutrition Examination Survey, which conducts measurements in blood and urine samples taken from a representative sample of the population. And what he found was that greater than 25% of a group of about 330 measurements that he looked at strongly correlate with household income. And this included microbiological factors, nutritional factors, a variety of environmental exposures, physiologic, red blood cell counts, all sorts of things. So it gets in the body, right? What happens in our social environment is actually biologically measurable. And I also want to draw attention because it's actually been 25 years now since the term environmental justice was coined by this really brilliant article by Bullard and Wright in 1993, in which they really document how polluting industries focus on being cited. It's not just that poor people are the only ones who can, the only place that poor people can afford housing, afford to live is near polluting industries. It's the other way around also, that those industries purposely cite their dirtiest industries in the communities that have the least political capital and the least ability to fight back and resist. But that changes too. So then I'll talk a little bit now about our study, Childhood Autism Risks from Genes and the Environment, the CHARGE study, which we launched in 2003. And the goal was really to identify broadly the causes and contributing factors for autism and other developmental delays. And our focus was on environment and also the way in which genetic susceptibility might interact with environmental factors. And we cast a very wide net, and we've published on everything from pesticides and folic acid to diabetes and maternal infections in pregnancy. But I'll talk about the design here and then our work on air pollution and also gene-environment interaction. So the design is we have three groups, two groups of you can think of them both as case groups in relation to a general population sample that was matched to the ASD cases on age, sex, and geography. And the first two groups are, we recruit those through the California Department of Developmental Services database, which is a pretty unique feature. There are not very many states where there's this pretty strong, it's not exactly a registry, but it has very wide coverage. And we work with that department, and they've given us the ability to call those people. And then the birth files is how we get our general population controls. And then these are the eligibility criteria. And all of the diagnoses get confirmed in the clinic by trained, research reliable clinicians. So when we started the study, we sort of looked at, what was the existing literature and what factors are neurotoxins, and kind of put together this list: metals, organic pollutants, viral viruses, medical procedures, and nutritional factors, and then different ways in which we might be able to measure them. So we can look at residential history and link to databases, exposure databases, and of course that's how we do the air pollution. Metals we can measure in various types of media. Newborn blood spots is a way for us to get at like prenatal kinds of exposures. Teeth can also help us do that, and so forth. So the viruses, there is a very substantial literature on congenital rubella, when there was an epidemic in the late 1960s, and there were odds ratios of about 50 for children who'd been exposed, and so forth. We're focusing on the period starting 3 months before conception, all the way through the pregnancy and up to the first year of life, and we also have data going further forward. This is just to emphasize why there are so many studies on air pollution and autism compared to a lot of other things, and that is that we do have a lot of monitoring networks that have been put in place by EPA, and in some cases states have augmented those. This just happened to be August, and you can see California had some pretty bad air pollution during August, as we all experienced, from fires everywhere. And I want to say that although air pollution from vehicle exhaust has been the primary source of air pollution in the U.S. for several decades now, it's now going to be vying with smoke from wildfires as a primary source of air pollution. In this study, we collected their residential histories. We mapped the homes and derived two metrics. One was distance to freeway, which gave us a sense of their traffic-related air pollution, and then the second was using estimated concentrations from monitoring of regional pollutants, including coarse and fine particles and NO2 and ozone. And what we found was that those mothers who had been living during their pregnancy in the closest 10% of our population to a freeway, which was less than 0.2 of a mile or about 300 meters, had an odds ratio of about 1.9 relative to those who were in the furthest 50% of our study population. And that those models were adjusted for various sociodemographic factors, as well as maternal smoking. And then the other analysis was with the regional air pollution estimates, and these are just a few of them, but you can see that with the fine particles, there was a pretty strong association for the first year of life as well as during the pregnancy, and slightly weaker during the earlier you went during the pregnancy, but still some suggestion of impacts. I'm not going to dwell on biological plausibility. You've just heard a beautiful, elegant presentation of things I didn't even know about. But I do want to emphasize the first item on this slide, which is that you might have noticed that there was not a dose response. So those middle categories of the 10 to 25 and 25 to 50 percentiles of distance from the freeway did not show any elevation. And that actually turns out not to be unexpected, because in fact the particle concentrations, of course, they're highest closest to the freeways, and by the time you get to 300 meters they're back to background levels. So in fact the shape of the dose response that we saw is exactly what we would expect based on dispersion. So this is the broad account of what we have in the literature. There may be some studies I've missed here, but I think I've got pretty much all of the original studies. There have been several meta-analyses that have also been done, concluding that the evidence was limited. Now, one of the problems in these studies is that they don't all measure the same things. So when somebody tries to do a meta-analysis, they might find 3 or 4 studies that actually have the particular, PAHs or solvents that they're looking for, and some of them looked at criteria pollutants, and others more at the air toxics. Nevertheless, the vast majority do show associations with autism based on prenatal exposures, and the 2 salient ones that really don't see very much of a signal or any are both from European cohorts. Which kind of raises some questions about, what are they doing that's different from what we are doing, and the air pollutants are probably not that hugely different in terms of the composition of their air pollution, although there are some differences. The one thing that was interesting is that most of these were looking at a diagnosis, and they varied. Some of them are self-reported diagnoses. For example, in the Nurses' Health Study, they used self-report. On the other hand, in the ROS study, 2015, it's about 2/3 of the way down the slide, they did a validation of that, and one of our speakers for the afternoon, Mark Weisskopf, he'll be talking about later life stuff, but he's also worked on this, and they did validate that the self-report was a very good, very reliable measure, and even those few who didn't meet criteria for autism when they went back to records were actually very close. They might have been 1 point short of actually having the diagnosis. I think ours, which is the Volk et al. Studies, I think ours might be the only one that actually did a full clinical assessment of the children. But again, we also found that the vast majority of children from the DDS, the Department of Developmental Services database, did meet criteria or were very close to meeting criteria. The two European studies did not actually use diagnoses. They use continuous quantitative traits of autistic behaviors. Although the Gong study then used that screener to then divide people with a cut point, but it's really a screening instrument, not a diagnostic instrument. So there may be some issues about the quality of the outcome data in those studies. But the other potential explanation may be, and going back to this multifactorial model, that there's different cofactors operating in the European setting right now. And Europe has actually banned about 200 chemicals that we are still exposed to here in the US. So I think we really need to look at these interactions as being potentially relevant to the inconsistencies in the data. Causal inference, I think most of these studies did a pretty good job of adjusting for various types of socioeconomic factors, race, education, income. Nutrition, not so much. I think that's not as easy to get as socioeconomic variables. And then there are seasonal factors that could play some role. Again, maybe as cofactors, not to say, in other words, it could be confounding, but it could also be heterogeneity. I mentioned the potential for misclassification. I think the exposure variables in pretty much all of these studies were really very meticulously assembled and put together and based on the best available data and models that are out there that have been validated. And then the other thing, though, that makes me pause about the issue of air pollution and autism is that the cigarette smoking data is not, there's just very little support at this point. Now, they're not great studies, most of them, but people are not finding an association between cigarette smoking and autism, and that's a little bit disturbing because the overlap of chemicals in cigarette smoke and vehicle exhaust or air pollution is pretty enormous, thousands of chemicals in common. So I think that's a question that needs to be resolved to really have coherence to the story. And we are in the ECHO project, which is the Environmental Influences on Child Health Outcomes. It's a national synthetic cohort bringing together, harmonizing cohorts from around the country of close to 50,000 children. We have a project involving some of those cohorts right now, and we're going to be trying to take a better look at the cigarette smoking and autism connection in that context. So I want to emphasize, though, that this inconsistency is not necessarily an argument that there's no causal association. It may well indicate true heterogeneity, and that's something I think is not thought about a lot when we evaluate evidence. In the world of autism, a great emphasis has been placed on genetic factors, and in fact, if you look at the funding, it's about 95% of the research on causative factors has been in the area of genetics and 5% on environment, and that has not changed in the last 15 years. And the model on the right of the, dividing the pie into two camps, it's either one or the other, has also been a bit of a problem in the way in which heritability has been described. On the left, I'm suggesting that more than half, maybe even 90% of autism may in fact be related to overlap of genetic susceptibility, if you're thinking of it in relation to environmental factors, or environmental susceptibility to the genetic factors if you happen to be a geneticist. And we took a look at genetic susceptibility using, we've done it for a few factors, but for this project we looked at copy number variation. And so this is a slide showing copy number duplications and deletions. So along the first few rows there, you can see the orange box is a segment of the DNA that's been duplicated 2 times in one row, 3 times in another, and then the next row shows kind of a duplication of that, the light blue, dark blue segment, a larger segment that's been duplicated. And sometimes these copy number, sometimes there are hundreds of duplications of a segment of DNA. And then sometimes there are sections that are missing. And so we constructed a couple of variables that were the sum of the lengths of all of the deletions and the sum of the lengths of all of the duplications as a measure of global burden of genomic instability. And from that, we then looked at cases versus controls, autism versus typically developing TD children. And what you see is on the right, most of the autism cases tend to dominate the right side of the slide with the higher burden of larger amounts of duplicated DNA. And on the left, it's more the typicals. And then this was the analysis we did looking at gene-environment interactions. So there's 3 panels here. The top are the deletions, the middle are the duplications, and the bottom panel is the total. And then we've got these 5 different pollutants. And then for each one, we looked at the 3 time periods shown on the left, the pregnancy, the first year, and the second year. And then the blue represents the environmental odds ratio, the red represents the genetic, those are main effects, and then the purple is their combined effect. And I think if you look right in the smack in the middle, ozone and duplications, you see very strong evidence of an interaction, a synergistic impact of the two. So the children who had greater genomic instability, inherited or de novo, some of it could have been, and had the higher ozone exposure were at exceptionally high relative risk odds ratio for autism. There's different ways to measure genetic susceptibility, and more people are looking not just at the code itself, but also at epigenetics and gene expression. And then we, in the environment, we have what we breathe, what we eat, what we drink, medications we take, products, household products, and personal care products. And the important thing here, of course, is that these are what provide us with the public health way to intervene. And going back to those causal pies, think of it that if you can intervene, you don't have to intervene on everything. You can intervene on one component of a sufficient set of causes, and it's no longer sufficient. You might have someone with some impairments, but not having reached that threshold of a diagnosis. So gene by environment interactions have really been rarely looked at, actually, in this field, despite lots of environmental factors and lots of ways in which genetics has been looked at over time. The heritability estimates are always given as very high. I'm told that it takes about 20 years for people to stop quoting figures that, statistics that are no longer hold. And so we probably have a few more years to go of still hearing people say it's 90% heritable. But the very large, I will say those earlier studies were much smaller studies, and these were some really huge studies that are below here. And the Sandin study looked at thousands of people and looked not just at, monozygotic twins, dizygotic twins, siblings, cousins, and so forth, and came up with estimates of the heritability somewhere in the, around the 50% range. These are everything there is on gene by environment interaction in autism. The first two are actually just sort of discussions that should be looked at. And then there are these three sets of studies that are actually all from the CHARGE study, looking at synergies between prenatal vitamins and one-carbon metabolism genes, between the MET-C gene and air pollutants in the third trimester. So that's another one that came out of CHARGE. And then the one that I just showed you, the Kim burden of CV. There are two studies that looked at were case-only studies, and so they looked at the severity of autism, and one of them showed that copy number variation plus maternal infection, and the other one, copy number variance plus ultrasound in the first trimester, was associated with greater severity of autism phenotype. So that's kind of the synergy approach to gene by environment interaction. There's a growing literature, I do want to say, on air pollution and stress. I've seen many papers now on reproductive outcomes, and Tanya Kemet, who is my graduate student who is here, I don't know where she's sitting. She has been doing her research on economic hardship as well as mental health of mothers, and in combination with air pollution and some of the CalEnviroscreen metrics of vulnerability. And you'll be seeing things from her soon. And nutrition has also been looked at as a buffer against environmental factors. So this is the synergy model. There are other models. We can look at environment in relation to gene expression, to epigenetics, metabolomics. The bad news is we've still got thousands and thousands of chemicals out there not tested for neurodevelopmental toxicity, and we have lots of evidence showing that pregnant women and newborns are at birth already have a lot of chemicals in their bodies. But the final thing I want to say is multifactorial causation is more than a concept. It is actually a compass to action. And this is the slide that really tells us that all you need to do is act on a few of these components of sufficient causes that may be a part of multiple sufficient sets. So this is, the B factor here, whatever that happens to be, and it's in all three of these, and it might be in 5 other sufficient sets of causes. That's where we can intervene and actually be impactful. And so then I need to thank my collaborators, co-investigators, and many, many staff who've contributed, a couple thousand families now in CHARGE, and our funders. And thank you.